Okay, well, good morning. My name's Lee Smith. I'm from Greyrigge Associates. We're a technical consultancy based or headquartered in the UK, but we have offices in Cambridge, Massachusetts, and also in Japan, in Kobe. Just to give you some background really on who I am and what we do, we work across all modalities, so cell therapy, gene therapy, vaccines, antibodies, mRNA, helping organizations, startups, mid-sized companies, multinationals in developing products through the clinical phases, really from post-discovery all the way to commercial launch. Okay, so today I'm talking about application of quality by design in cell therapy development, and this is based upon a real-life example of how we work with a company that was a clinical phase company moving into phase 3 and really bridging that gap. Just to give some background on allogeneic cell therapy products, the question is, how do you transform a product that's from a donor that's heterogeneous with biological material that's reproducible and ultimately can be licensed as a therapy. So the context here is it's mesenchymal stromal cells. They're inherently variable. And that's based on the donor biology, the tissue source, the expansion potential. Okay, and MSC therapies, they rely on unrelated donors, and I'll explain that in a moment. So that brings additional biological and regulatory complexity. So the question is, or the core challenge is, how can we control this variability and also meet the regulatory expectations really for consistency from potency and safety? So what's at stake really is the patient's safety, the efficacy of the product, and also importantly, the batch-to-batch consistency. Can you make it, and can you make it consistently? So why do we need multiple donors and master cell banks for these type of products? Well, the MSC expansion, it's limited. You can only passage it so many times before you get senescence. So you can't expand things indefinitely. So with some of these products, there's high dose requirements, repeat dose indications. So you require large volumes of cells. So single donor manufacturing isn't sustainable. You have to keep on getting more donors through the life cycle of the product. So multiple donors are required to maintain supply. Okay, so each donor will actually produce a separate master cell bank. So we have to make sure that they're as similar as possible. So ongoing donor sourcing is required for the life cycle of the product. So we've got donors coming in and master cell banks coming out through the hopper, so to speak. So the donor selection for MSCs, it involves, you have to screen the donors. So looking at their medical history, looking at infectious disease testing, so performing a risk assessment that you can start to screen out essentially the right donors that are eligible. There's a physical examination, so you're really looking for signs of infection or chronic disease, looking at their BMI, looking at their health status, and their age range. Typically if you have younger donors, the cells actually behave better or more reproducibly. But if there's any history of transmissible disease, they will be screened out. So once you've got your donors, and let's say we start with 100%, by the time we actually get down to those lead donors, we're looking more at 10 to 20%. So you're screening out the vast majority of the people who try to donate. The donor selection, I won't go through this laundry list of infectious disease testing, but things like HIV, syphilis, Zika virus, etc. And you're looking for things like mycoplasma infection, endotoxin issues. So donor-specific variability, the problem is really these unrelated donors, they all have differences in their cell behaviors. So the immunophenotype is different, the growth kinetics can be different, proliferation potential, cytokine secretion properties, and these are all influenced by the donor age, the sex, the ethnicity, and also how you harvest the tissue. So there are lots of things that can cause variation. And this impacts on the manufacturing and also the regulatory aspects of this. So this culminates really in batch-to-batch inconsistency or consistency if you get it right. So then this really complicates the critical quality attributes of the products and the critical process parameters. So this really drives the need for that rigorous functional testing to select those lead donors. Okay, so, and the assessment really, you'll be looking at growth, identity, differentiation, immunomodulation. So there are going to be certain factors really that you're going to pick the sales on. So if we really look from taking a donor through to producing drug product, there are essentially 11 steps. So we have the donor selection and screening. We have the tissue collection. Okay, we have the cell isolation and enrichment. And then we have the initial cell expansion. And that's really trying to move to your master cell bank. So the characterization and the QC testing that I just mentioned before you produce your first master cell bank. From that master cell bank, there will typically be a working cell bank. So that will be expanded to make the product, so that your large-scale cell expansion. And then there will be a harvest and a formulation. Obviously, then you have to test that. And then finally, you're going to freeze these down and cryopreserve these. So those are the 11 steps really going from donor to drug product. And this is required throughout the life cycle of the product. So if we start to look at the process flow, this is really where things start to get complicated. So here for instance, is what the process unit operations, and you can see the unit operations on the top and the bottom, and all of the different process parameters actually live within those unit operations. Many are similar, but obviously you're at different stages, so everything needs to be considered. And there are many, many moving parts in this whole thing. So if we're mapping the process, we really start with the unit operations. So again, you can see those really to filling the product. And what we need to take into account really is, what are the quality attributes of the product? I mean, for instance, an easy one would be viability. Okay, are the cells viable? Okay, what is that viability, or how many cells have you got? That would be an obvious one. So we're looking at those outputs, and those outputs, those responses, those quality attributes are going to be tested at each unit operation. Okay, and with this particular process, there wasn't a drug substance, so you're really going from donor all the way to drug product. Okay, and that's what you're releasing. Okay. Of these outputs, not all of them are critical. Some of them are, many of them are, okay, but many of them aren't. Okay, but again, you don't know that because you're developing the product and it's evolving. And then if you look at the inputs, again, that you can see from the previous slide, many moving parts. So how do we actually try to identify of those attributes which are critical? Because again, you want your product to have the efficacy and the safety and the identity and purity. How do we work out really of those inputs what are critical, what will impact on those critical quality attributes, those critical process parameters? So if we look at the product lot release testing, in this particular case, identity, so using flow cytometry, looking for CD markers, both positive and negative in the cells that we're expanding, and again, they can change. The purity and safety, so looking at things like sterility, mycoplasma, endotoxin, adventitious agent testing. So you're performing this fairly standard set of tests. The potency, so immunomodulation, cytokine assays, cell viability, cell count. And then general tests, so visual inspection. So looking at color, looking for particulates, container defects. And then finally, really, the process-specific impurities. So if you're using trypsin, does your downstream process actually remove the trypsin? Does it remove any residual FBS? So common causes of lot failure with these types of products under safety, contamination. Okay, that's an obvious one, and it does happen. And good GMP and good techniques will actually limit that. Potency, low cell viability, again a very common one. Or maybe potency failure, so T-cell inhibition. And things like the flow cytometry profile that I mentioned before, or you've got cells that aren't the MSCs in there, so contamination. So high trypsin, high FBS, and particulates by VI. And I flashed that one because this can be a particularly pernicious problem in cell therapy, and it needs to be handled very carefully. but it's probably a separate discussion on its own. So what we're really trying to do is manage that variability of those donor-derived MSCs to make a standardized culture. Okay, and we're trying to improve the product quality, we're trying to reduce that batch-to-batch variability and bring regulatory compliance. So the things that we'll be looking at would be the media and the reagents, the passage number and the seeding densities, so maximum defined passages, controlled seeding densities, things like that. Environmental controls, so that could be CO2 or oxygen tension, and harvest and expansion parameters. Okay, and you can see on the left, I've got a fairly typical profiler of knowledge space. I'm going to go into that in just a moment. But what we're trying to work out of all of these conditions, from all of those inputs, what's critical, what really matters, what matters the most. So we can apply quality by design for cell therapy. Okay, so the traditional approach really for development of products, and not just cell therapy, but most modalities is, you'd lock the process. Okay, so you'd validate the process, you'd lock the process, and you wouldn't change it. Okay, so you would run 3 PPQ lots, you'd validate it, and the process is fixed. Okay, and thou shalt not change it thereafter. Okay, with the enhanced approach, quality by design, it's described as enhanced approach, which is based on ICH-Q11. Okay, so that's the development and manufacture of drug substance, okay, for biologics, or biopharmaceuticals, should I say. And that's the QbD approach. So you're working on a design space to give you flexibility with the regulators, but also with your manufacturing. Continuous process verification, so once you've actually got your process under control, it's evolving, because again, you're making relatively few batches at the beginning as you move to a commercial launch. So you want all of that data, because there's sparsity of data when you're developing a product. And it's really building up that control strategy with process flexibility. So we need the product CQAs. Okay, we try and define these as early in the development process as we can, but it's not easy because it's based upon preclinical or clinical information. So you're trying to link these things, but you can use prior knowledge, prior data, experience. Okay, so you can have educated guesses at the beginning. But the CQAs, they would affect safety and efficacy of the product. So if we start with the QTPP, so that's the quality target product profile, which we derive from the target product profile, which considers more aspects of dosing, of storage, of commercial aspects. So we have the QTPP, and we'll be considering safety and efficacy aspects of that, such as identity, potency, purity, safety. Okay, so we're breaking that down and looking at our potential quality attributes, and we're performing a criticality assessment. But ultimately, will culminate in the CQAs. Okay, and this will happen over time. So to put together a process control strategy, and this is a relatively complex diagram, but stay with me. What we're trying to do, based upon that QTPP, is put together a parameter attribute matrix, okay, or a PAM, okay, which is ostensibly it's a risk assessment. So you're taking all the information that you have about your process at the time that you have it, the attributes associated with the product, and you're risk assessing it. And I'm going to come on to how we apply this. And then you're really taking all of these parameters, because again, you saw the inputs, there are hundreds. Okay, so you can't look at everything. Okay, you've got finite resources, finite time, finite money. So you have to make some choices based upon education, prior knowledge, prior data. So what we would do next, really, once we've actually identified and narrowed some of these things down that we want to investigate, we would perform DOEs, so screening experiments. And what you're trying to do there is identify those things that are important and those things that aren't important. Once you've identified those, you can then go into optimization of those and verification. Okay, so once we've done that, you've got a model for the process. Okay, it's just a model though. So what you have to do, you have to perform robustness testing. So when you start making this at scale, you will start to rattle the cage, so to speak, to see, okay, does this process stand up? Is the model actually correct? Okay, and that's where we get into proven acceptable ranges, normal operating ranges, how you're going to run this on a regular basis. And the terminology around that is design space and control space. And then simply after that, once you've confirmed the robustness of the process, then you're actually going to run that process routinely. Okay? You're going to build up more information, but everything should fundamentally live on what you've developed there in terms of your process control. You understand what's important, you understand where the optimums are, and you're running this on a routine basis. Okay, and that's a continuous risk assessment and improvement. Okay, so those are the different stages really. You go from a paper exercise, risk assessment, you go through experiments, and then obviously you start to perform this on a routine basis. So the parameter attribute matrix, we have our list of critical quality attributes. Those attributes such as cell viability or immunomodulation. We have our list of process parameters. We don't know which ones are critical at this point. So we adopt a standard FMEA approach. So severity, occurrence, detectability. Okay. So again, nothing new here. We're taking existing approaches. And what this will produce is a prioritized parameter attribute matrix. So this is what our parameter attribute matrix look like. We do it in Excel. You can use software. There's proprietary software available that some organizations use. But what you can see on the left really are the list of the process parameters. On the top, you've got your CQAs. In this case, we've categorized them into safety, into efficacy, and into commercial. So commercial could be meaning the cost of goods. And you can see really within there, you can see different shades. Okay, and that's really starting to show your hotspots, okay, of your process where things are being impacted. So if we zoom in on that, okay, what you can see really on the left, we've got our unit operations. So for instance, it could be the thaw step of the process. Okay, we have the parameters associated with that, and we have the ranges that are currently set. Okay, they may not be optimal ranges at this point. Okay, and that's what you're doing. You're going through the process. Then we have the CQAs and the particular quality attribute that you're interested in. And you have to score this. So, you have to score this. So it can be 1 to 10, it can be 1 to 5, it can be 1 to 3. I mean, it's really up to you. But you're really using subjective information and experience to try and identify those things that are important that you want to take through to the next step of experimentation. And that's the risk of impact. So once you've done that exercise, you can produce, in this case, a Pareto chart. So that gives you the order of things that, by your assessment, are the most important. Okay, so, and again, you can weight them if you want, because safety obviously is absolutely critical, but based on efficacy as well, and cost of goods. You can weight them equally, you can distort them. And there's a documentation trail that exists with all of this exercise. That's important. Again, you're thinking not only internally that you need this, but also for regulatory aspects. So you start with your TPP, you derive a quality target product profile, quality attributes associated with that, specifications, and you've got historical data. So historical data can give you information around your process and your process parameters, and these really come together in the risk assessment or the PAM that I just described. And you're asking a question, okay, does this parameter impact upon the quality attributes? Okay, and the answer will be yes or no. Okay, so if it's a yes, it's a potential critical process parameter, something that affects the CQAs. If the answer is no, then you may ask, okay, does this affect the yield or the productivity of the process? And if the answer is no, it's a non-critical process parameter. If the answer is yes, it's potentially a key process parameter. And those two come together really to form your characterization plan and the basis of your control strategy. And associated with all of these, there's documentation. Okay, so there's a documentation trail that you're building over time that you're going to use in your dossier submission. But you'll also be using that when you're interacting with the regulators. Okay, so as I said, what you're trying to do here is avoid reinventing the wheel. Okay, so you're trying to use that prior knowledge, external knowledge of similar products. You've also got your internal historical data that you could use. Plus, you're going to use that to support your new experimental data, the design of experiments that you perform. Okay, so all of this really comes together to form the basis of what you're doing. So there's nothing wrong with using existing data, other people's information, publications, literature, but also the data that you develop yourself. And this is really just explaining the basis of the quality by design. So PARs and NORs. So that's your proven acceptable ranges and your normal operating ranges. So what you can really see is on the left, you can see the knowledge space. And that's when you're developing your process, it's all the knowledge that you have about it, when it fails, when it succeeds. But it's really trying to, obviously you don't want failure, but you need to know where the edge of failure is. So you're building that design space, okay, where you can operate your process and your CQAs will be met. The viability will be good, the immunomodulation will be acceptable. Okay, and within that design space, you have your control space. Okay, so you're really trying to control the process more tightly. Okay, so if you look on the right-hand side, you can see the process. So on the top, you can see 3 different colors. You can see a red, an amber, and a green. Okay, so that's really your knowledge space, your design space, and your control space. And if you're actually within amber or green, your CQAs are met. Okay, your specifications are met. Okay, and that's the approach that you take with quality by design. Okay, so if we look at the product development phases for cell therapy, but this is really a template that can apply to many modalities. Obviously, from preclinical through to commercial, we get increasing process knowledge. Okay, so increasing CMC knowledge. Very often, the process scale will increase over time. So you start small and you build up. And your control, really preclinical to phase 1, that's where you're performing the development. Okay, you're refining the process after phase 1, and you're beginning to optimize it. You have to bring it under control. It has to be productive. Okay, it has to be in control. And then ultimately, you're going to validate it, which will demonstrate the control. And the GMPs, you're applying phase-appropriate GMP. Okay, so at the beginning, R&D, preclinical, there's no GMP. Then you apply early phase GMP, which is a bit more relaxed. Then ultimately, you go into full GMP, really. Typically, in our experience, really, from phase 2 onwards. And the DOE sweet spot really sits between phase 1 and phase 3. Okay, that's where we perform it, because things are developed enough. You have to have the skeleton of your process before you start to optimize things. Okay, so and what you're really doing through this exercise, you're building regulatory agency confidence. Okay, so you're linking the CPPs to the CQAs and demonstrating they're critical. Okay, you're presenting a comprehensive risk assessment and a lot failure mitigation strategy. You're showing evidence of improved process control and consistency. And it's really a data-driven narrative, that the product's safe, it's efficacious, and the manufacturing is ready for a BLA. Okay, that's really where you're going to. So you're proving it to the agency, as well as actually getting those benefits internally as well. So in this particular case, the challenges that we saw with this organization is that they had no TPP, they had ill-defined CQAs, no CPPs, they had no process limits or poor process limits. Their cell-based assays were poorly optimized or not under control, and they had phenotype variation in the CQAs. Okay, and this set them back at least 18 months, longer to be frank. So what we did when we started to work with them, we really started to bring things under control, both around the assays and the process. So performing these risk assessments and putting the statistics and the DOEs together really to bring the process under control, okay, and making the process robust and rugged. And the positive impact is, there's a greater understanding of the assay and the process variants. We're driving the CPPs and the CQAs and documenting everything, and really just getting a better understanding of the process and the product. So the application really, we're performing risk assessments both on the assays, so DOE on assays, and also the DOE on the process. Okay, and some of this work actually, in a holistic sense, it really started to help as well in terms of some of the clinical considerations when you start to look at different markers, okay, because of the nature of these type of cell therapy products. So the continuity plan really is that we're defining the TPPs and the QTPs, the CQAs, the CPPs, etc., performing those risk assessments on the process, also on the materials, on the assays, and also on the clinical. Okay, and we're driving the understanding and we're putting mitigations in place. And using statistical tools, and that's important to get the statistics right when you're involved in doing these type of exercises. So with allogeneic therapies, so this improves the comparability, the characterization, especially product batches from different cell banks, where you've got that inherent variability. But it's really integrated thinking. You're bringing the process, the product, the clinical, the assays all together in one sort of confluent process where you're taking all aspects into consideration. So the strategic benefits of process understanding: a process is optimized, and you identify those CPPs to run a leaner, more reproducible operation. Okay, you get less lot failures, lower batch rejection, lower deviation rates. It's IND enabling. Okay, smoother regulatory filings. You've got regulatory confidence. You've got the agencies have confidence in you that you know what you're doing, and you can put together a really strong CMC package. It can also help if you're working with CDMOs. It's faster tech transfer because the CDMO is confident that you know what you're doing. You understand your process. Okay, so that actually builds an improved CDMO relationship because, again, nobody wants failure. So the takeaways really from this, I mean, the donor variability must be managed. It can't be ignored. The CMC transforms these variable MSCs into a reproducible therapy. The QbD adds value, even if adopted late, and that's one of the main points here. In this case, this company was in phase 2. It hit delays because it didn't apply these techniques. Okay, but again, once these were applied, it really got things moving. And the risk-based thinking strengthens those late-stage CMC packages. So I wouldn't recommend not doing this. I wouldn't wait until you're heading into phase 3 and discover this. The earlier you adopt it, the better. You're having a plan, and the process and the analytics should evolve together, because if your assays aren't in control, how can you read your process? And this continuing alignment, it ensures a robust control and regulatory success. So, and a robust process. So if these issues sound familiar, then happy to have a chat. We work with companies that are startups, but also as in this case study, products that are heading into phase 3. And if you see any of these items here that you keep you awake at night, then as I say, happy to have a chat. So thank you for your attention, and happy to take any questions.
Thank you, Lee. Does anyone have any questions?
Good, all clear.
Yeah. Oh, one here.
I have a question, please.
You have a question?
Here.
Oh, yes.
So any clinical outcomes that have translated to your process in any way, can you give us an example of that?
I can't go into too much detail on that, but because of the nature of some of the potency assays, they had a direct translation. So for this particular product, by building this understanding, particularly around the assays, there are multiple assays, and you're really trying to work out with these type of products what's important. But some of these actually carried over into the clinical as some of the markers they were actually looking for in the patients for this particular indication. So it does have application. It's something that I see more in cell therapy than I see in other modalities.
Great,
but I think it's the nature of the product.
Do we have any more questions? In that case, can we thank Lee one more time?
We've got one here, I think.
Sorry.
Very few cell therapies actually get approved. What do you attribute that to?
I think in this particular case, it was they were suffering major CMC issues. And I think with many products, CMC is often a neglected aspect of it. I think if you look at cell therapies, the process is complicated as any, whether you're producing a monoclonal or a gene therapy or a viral vector, it's the same fundamental issues. Okay, so in this case, what was actually causing the issues on the delay with the agency really was this complete inability to manage that process. So they're ending up with repeated lot failures. Once they brought that under control, which took about 2.5 years, then the agency was very receptive, and this product actually got licensed, I think, about 3 years ago. Thank you for your attention.